Okay, we are finally to chapter 29, the last chapter in A&P. Congratulations, everyone. You've made it to the end. You should be super proud of yourselves. This is a challenging course. This material takes a lot of time to digest and really think about the bigger picture behind the things that you're learning. It's not just straightforward memorization, although there is a lot of that involved too. So this is a hard class, and you should pat yourselves on the back for coming this far. In this last chapter, we're going to talk a little bit about development, that is embryology. We're going to talk about pregnancy, talk about prepartum and postpartum changes that happen to women during pregnancy, and then we're going to get into a little bit of human genetics and heredity. All right, so the period of development from fertilization up until birth is going to be called the prenatal period. It begins with fertilization when the secondary oocyte and the sperm unite, and it's going to end about 38 weeks later with birth. The prenatal period is composed of three shorter periods: the pre-embryonic period, the embryonic period, and the fetal period. First, there's the pre-embryonic period. This is going to be the first 2 weeks after fertilization occurs. What happens here is the single cell that's produced by fertilization, which is called a zygote, becomes a spherical multicellular structure called a blastocyst. And then that blastocyst is what implants in the endometrial lining of the uterus. And when that happens, the pre-embryonic period ends. Next is the embryonic period. This is the weeks 3 through 8. Rudimentary versions of the major organs are going to start appearing here and starting to develop. And now we call the organism an embryo. After week 8, the remaining 30 weeks prior to birth are going to be the fetal period. And this now we call the organism a fetus. And the fetus continues to grow and increase in complexity during this period, and the fetal period ends with birth. Now, when we talk about embryogenesis, what we're going to be talking about is the developmental period of the pre-embryonic and embryonic periods. All right, first, the pre-embryonic period is going to begin with fertilization. And remember, this is going to be when two haploid gametes fuse to form a new diploid cell, and we call this single cell the zygote. Now, the zygote is going to contain genetic material from both mom and dad. Fertilization is going to restore the diploid number of chromosomes, right? We're going to go from the 23 chromosomes in the gametes to 23 pairs of chromosomes in the zygote. Fertilization is also going to determine the sex of the organism, whether or not the father provides the X chromosome or the Y chromosome is going to determine the sex of the organism. Fertilization is also going to initiate cleavage. We'll talk about that in a second. It's going to occur, remember, in the widest part of the uterine tube, the ampulla. Now, the oocyte remains viable for 24 hours following ovulation, while sperm can remain viable in the female reproductive tract for up to 3 to 4 days. So there is a kind of a window of time where one can have intercourse, and potentially that can lead to pregnancy. Before fertilization can occur, a process called capacitation has to happen. And this is a physiological conditioning that sperm undergo that basically makes them capable of fertilizing that secondary oocyte. Capacitation is going to occur in the female reproductive tract. What happens is sperm normally have these protective glycoprotein coats and some proteins as well that help protect them in the male reproductive tract. These get removed from the sperm in the female reproductive tract, and this is going to enable the sperm to make contact with the zona pellucida and penetrate it and fertilize the egg. Okay, sperm, there's going to be millions of them that get deposited after ejaculation in the female reproductive tract. About a few hundred are going to make it all the way to the secondary oocyte and get a chance at fertilization. They're going to be attracted to that oocyte by chemicals that it releases. For example, progesterone is released by the cumulus oophorus cells surrounding the oocyte, and progesterone is going to bind sperm, causing an influx of calcium into that sperm. And this changes the swimming patterns of the sperm, and they swim toward that oocyte. When they get to the secondary oocyte, they're going to attempt fertilization, and only one sperm is going to be able to fertilize. Right, there are actually 3 phases of fertilization. The first phase is corona radiata penetration, and remember the corona radiata is that layer of granulosa cells surrounding the zona pellucida, which surrounds the secondary oocyte. So the corona radiata is the outermost layer. Then there's zona pellucida that the sperm have to penetrate, so that's the second phase of fertilization. And then the final phase of fertilization, once a sperm is able to penetrate that zona pellucida, it's going to fuse its plasma membrane with that of the ovum. So that's phase number 3. So the plasma membranes fuse, and the chromosomes from the ovum and the chromosomes from the sperm join together and form in a single nucleus. All right, first up, corona radiata penetration. This is the first phase of fertilization. What happens here is sperm are going to reach the secondary oocyte, and they're going to initially be prevented entry by that corona radiata. And underneath that corona radiata, remember, is the zona pellucida. Now, several sperm can push through the cell layers of the corona radiata. So several sperm can reach the zona pellucida. Now, the second phase of fertilization is going to be zona pellucida penetration. When sperm get to the zona pellucida, it's going to trigger this thing called the acrosome reaction. So species-specific proteins on the zona pellucida itself are going to bind to sperm, and this triggers the acrosome reaction in the sperm, and it releases these digestive enzymes, which allows that sperm to penetrate the zona pellucida. After the sperm penetrates the zona pellucida, immediately after, there's going to be an immediate electrical change and hardening of that zona pellucida, and this is going to prevent other sperm from being able to penetrate the zona pellucida. And this ensures that there's only one sperm that fertilizes the oocyte. If this mechanism fails, you get a condition called polyspermy. This is where 2 sperm enter simultaneously, and it's an immediately fatal condition with 23 triplets of chromosomes instead of 23 pairs. It is not conducive to life. Okay, and then you reach the third phase of fertilization, and this is when the sperm and the oocytes fuse their plasma membranes, and the sperm and the ovum conjoin their pronuclei together. So what happens is you get the contact of the sperm and the oocyte plasma membranes. They immediately fuse, and the sperm is going to basically dump its nucleus into the oocyte. Now, some mitochondria from the sperm can enter in this process, but they're going to either be tagged for degradation or they entirely lack mitochondrial DNA. We're not entirely sure which. But the point is that no mitochondria from the sperm are really going to make it into that oocyte. They're not going to survive in the oocyte. So mitochondrial DNA is only going to come from the organism's mother. Now, when the plasma membranes fuse, this is going to trigger that secondary oocyte to complete its second meiotic division. Remember, it was arrested there in meiosis II, and fertilization triggers its completion of meiosis II, and it produces an ovum and a second polar body. Now you're going to have the nucleus of the sperm and the ovum, and these are called pronuclei at this point. Each pronucleus has a haploid number of chromosomes, and the pronuclei are going to fuse and become a diploid nucleus. And at this point, you now have a zygote, which is a single diploid cell. Okay, here we have phase 1 of fertilization. You can see the secondary oocyte here is arrested in metaphase 2 of meiosis II. Here's the first polar body that it made after meiosis I. Here is the zona pellucida. Remember, this is a rich protein coat surrounding the oocyte that protects it and triggers the acrosome reaction in sperm. And then further outwards is the corona radiata, that single layer of granulosa cells. Now you can see the sperm here are penetrating the corona radiata. Then you're going to get one sperm who's going to reach the zona pellucida first. And that's going to trigger the acrosome reaction. And then that zona pellucida gets hard, and it becomes impossible for the other sperm to penetrate it. Now, phase 3, you're going to get the sperm and the oocyte's plasma membranes fusing, and this is going to trigger that secondary oocyte to complete meiosis II. So now you have an ovum here. You have two polar bodies, right? One from meiosis I, one from meiosis II. And you have a pronucleus of the ovum. Here is the sperm's pronucleus. It emptied it into the oocyte at the time its plasma membranes fused with the oocyte's plasma membrane. Then these pronuclei are going to fuse plasma membranes, and the chromosomes in here and the chromosomes in here will reside within a single nucleus, and at that point you have your single cell, the zygote. Next, the zygote is going to undergo a series of mitotic divisions, and this is called cleavage. And I want you to know that cleavage is going to increase the cell number, so we're not going to be working with a single-celled organism anymore, but it's not going to increase the actual overall size of the organism. Size is only going to increase after the organism implants in the endometrium. Now, before the 8-cell stage, the cells are not going to be tightly bound together. They're going to become tightly compacted after the 3rd cleavage divisions. And this is called compaction, the process by which contact between the cells is increased to the max. Next in cleavage, you have the morula. This is the 16-cell stage. And the cells of the morula are going to continue to divide and divide until the morula develops a fluid-filled cavity called the blastocyst cavity. At this point, the pre-embryo is now called a blastocyst. And I'll show you a picture here in a second, but there's an outer ring of cells surrounding the cavity, and that's called the trophoblast. And that's going to go on to form the chorion or the chorion. And then there's the embryoblast. This is cells that are kind of packed within one side of the blastocyst. And it's the embryoblast that's going to go on to form the actual embryo itself. The chorion goes on to form the fetal part of the placenta. Now, the cells of the embryoblast are pluripotent, and they're able to develop into any tissue in the body. So if you plucked a cell from the blastocyst at this stage and grew it in a different medium, and depending on whatever chemical signals you gave it, theoretically it could become any type of cell in the body. Now, cells are totipotent, that is, they're able to develop into any tissue in the body and any extra embryonic membrane until about the 8-cell stage. But beyond the 8-cell stage, they lose that totipotency and they become pluripotent. All right, so here we have the zygote which contains the single cell with the polar bodies within it, which will degenerate, and it is surrounded by the zona pellucida. Then you hit the 2-cell stage. You can see those polar bodies are degenerating. The 4-cell stage, the 8-cell stage. Now, these cells are totipotent up until this point, and beyond here, they become pluripotent. So now we've got the morula, and remember, these cells are going to continue to divide and divide and divide until we get a blastocyst, which is this ring of cells surrounding a fluid-filled cavity. And then you can see the embryoblast here is sort of this pack of cells hanging out on one side of the blastocyst cavity. And that ring of cells surrounding is the trophoblast. Now the blastocyst, this is the organism that's going to actually implant into the endometrial lining of the uterus. All right, so here's fertilization through implantation. Here's the ovary. Here's that mature follicle releasing the oocyte. Okay, so ovulation here. Here we can see the secondary oocyte with the zona pellucida and the corona radiata surrounding it. Here we've now got sperm that have made their way up the uterine tube and are attempting to fertilize that oocyte. And you get one that makes it through the corona radiata and makes it through the zona pellucida and triggers the acrosome reaction. And then you also trigger meiosis II completion, and you ultimately end up with the ovum pronucleus and the sperm pronucleus, and then they are going to fuse together to form the zygote. The zygote undergoes a series of mitotic divisions until it becomes the blastocyst. And the blastocyst goes on to implant into the endometrial lining of the uterus. And this ends the pre-embryonic period and begins the embryonic period. All right, implantation. So by about the end of the first week after fertilization, the blastocyst is going to enter the lumen of the uterus. The zona pellucida surrounding that blastocyst breaks down, and the blastocyst burrows into the endometrium. This is implantation. The process of implantation begins by about day 7 and ends by about day 9. During this time, the trophoblast, that outer layer of cells, is going to subdivide into 2 layers, an inner layer called the cytotrophoblast and an outer layer called the syncytiotrophoblast. Now by day 9, after fertilization, the blastocyst is completely burrowed into the uterine wall where it contacts nutrients in the uterine glands. This is the beginning of gestation. The moment when the blastocyst implants into the uterine wall, that's when gestation begins. Human chorionic gonadotropin, or HCG. This is going to be produced by the syncytiotrophoblast, and it's a hormone. And not only does it signal the reproductive system that implantation has occurred, it's going to promote the maintenance of the corpus luteum within the ovary. And this is going to prevent menstruation. The corpus luteum, remember, is going to produce estrogen and progesterone, which thickens that endometrial lining and maintains a nice thick endometrial lining. And so promoting the corpus luteum and the production of estrogen and progesterone is going to maintain a nice thick uterine lining for the newly implanted blastocyst. Now, HCG is detected in the urine as early as the end of the 2nd week, and this is why it's the basis of most pregnancy tests. HCG is going to be high for the first 3 months of pregnancy, and then it's going to gradually taper off. This is going to cause the corpus luteum to degenerate, but by then the placenta is already producing its own hormones to maintain the endometrial lining and to maintain pregnancy. Now, even though the HCG levels do decline, they're not going to go all the way back down to pre-pregnancy levels until several weeks after the pregnancy ends. All right, here we can see implantation. So we have the blastocyst with the trophoblast and the embryoblast. Around day 7, we get the beginning of implantation, and you can see the trophoblast coming into contact with the endometrium. And that trophoblast has now become 2 layers, the cytotrophoblast and the syncytiotrophoblast. And these 2 layers make up the chorion. Now they continue to proliferate. In this chorion, this cytotrophoblast and syncytiotrophoblast are going to go on and contribute the fetal portion of the placenta. All right, chromosomal abnormalities and spontaneous abortion. So this occurs regularly during gametogenesis, fertilization, and even cleavage. If a chromosomal abnormality occurs and it's severe enough, it's going to result in a spontaneous abortion or a miscarriage. And that's because many of these chromosomal abnormalities are simply not compatible with life. Now, a lot of these spontaneous abortions occur within 2 to 3 weeks after fertilization, and that's usually before the pregnancy is even known. It's thought that about 50% of pregnancies are lost due to spontaneous abortion, and that's going to include pregnancies that are not even known. And half of these are going to be from just chromosomal abnormalities. All right, by day 8, what happens? Well, cells of the embryoblast are going to start to form these 2 layers, the hypoblast and the epiblast. And together, these 2 layers form a thing called the bilaminar germinal disc. Now, the hypoblast layer, this is going to be adjacent to the blastocyst cavity, while the epiblast layer is adjacent to the amniotic cavity. So here's the amniotic cavity. Here's the amnion. This layer here is the cytotrophoblast. This would then be the syncytiotrophoblast. And you can see it's penetrating the endometrium. Now, if we go back here to the amniotic cavity, we look, what's adjacent to it is the epiblast, this layer of cells here. And then on top of that layer of cells, or underneath it, I should say, is the hypoblast. The hypoblast is adjacent to the blastocyst cavity. There are a few extra embryonic membranes that develop, and these are going to be formed by the bilaminar germinal disc and the trophoblast. Now, these extra embryonic membranes protect the embryo, and they assist in nutrition, gas exchange, and waste removal. The first extra embryonic membrane to develop is going to be the yolk sac. This is continuous with the hypoblast layer. So right here, it does not store yolk like it does in birds and reptiles. Instead, the yolk sac in humans is an important site for early blood cell and blood vessel formation. The amnion, this is a membrane that is continuous with that epiblast layer. This guy right here. It's eventually going to enclose the entire embryo in a fluid-filled sac called the amniotic cavity, and it's specialized to secrete amniotic fluid, which bathes and cushions the embryo. And then there's the chorion we've already talked a little bit about. This is going to be the outermost extra embryonic membrane, and it's formed from that cytotrophoblast and the syncytiotrophoblast. Now, the cells of the chorion are going to blend with the functional layer of the endometrium, that thick endometrial lining, and together with that functional layer are going to form the placenta. The placenta is a specialized organ that develops during pregnancy, and it develops from portions of the mother's body and portions of the developing embryo. And it's the site where nutrient exchange occurs, okay, where oxygen is brought in and nutrients are brought in to the developing fetus, and wastes are removed from that fetus. All right, that brings us to the placenta. This is a highly vascular structure, and we said it functions as the site of exchange of nutrients, wastes, and respiratory gases. This is going to be between maternal and fetal blood. It also transmits maternal antibodies to the developing embryo or fetus. It's going to produce estrogen and progesterone. It takes over the role of that corpus luteum, which is degenerated. And so this estrogen and progesterone that it produces maintains and builds the uterine lining. And the placenta begins to form during the second week. All right, we said that the fetal portion of the placenta is going to develop from the chorion. The maternal portion is going to develop from the functional layer of the uterus. And then there's going to be a connecting stalk, and this connects that early embryo to the placenta. Eventually, it's going to contain the umbilical arteries and veins, and therefore be the umbilical cord. All right, the placenta has these chorionic villi, which are these fine finger-like structures formed from the chorion that kind of project into that functional layer of the endometrium, and they contain branches of the umbilical vessels. Now, gas and nutrient exchange. What happens here? Well, you have that functional layer of the endometrium that contains maternal blood vessels, and you've got the chorionic villi containing umbilical vessels. Now, it's important to know that the blood streams don't actually connect anywhere, okay? The maternal blood is not going to mix with fetal blood. What happens is that the blood streams are so close to each other that the nutrients and the gases can diffuse across. So you get O2 diffusing from the maternal blood into the fetal blood, and CO2 diffusing from the fetal blood to the maternal blood. Some characteristics of the placenta: most of its growth is going to be during the fetal period. It adheres firmly to the wall of the uterus. It's going to be expelled from the uterus shortly after the baby is born, and for this reason it's commonly known as the afterbirth. And it's a selectively permeable structure. It's going to allow some things to cross it and prevent other things from crossing it. For example, respiratory gases are going to pass freely. So are some antibodies. Microorganisms and some maternal hormones are prevented. Now, some microorganisms can cross the placenta. For example, some viruses, some bacteria, some drugs, alcohol, and several toxins. These harmful substances can, if they cross, cause birth defects or even death to the developing fetus. Dose and timing of exposure to these substances is going to affect what happens to the fetus. For example, some things with a small enough dose, the likelihood of having an adverse outcome is low. Some things, having the dose later in the pregnancy, for example in the third trimester, may result in no harm, whereas having the exposure in the first trimester may result in spontaneous abortion. Okay, so it all just kind of depends on the dose and the timing of exposure. Pregnant women are urged to quit smoking and refrain from taking drugs and using alcohol while they are pregnant, because so many of these substances cross the placenta. All right, so let's take a look at the formation of extraembryonic membranes, and this is early on in week 4. Let's kind of go from the center and work our way outwards. So in the very center here we have the developing embryo. Remember, this was the bilaminar germinal disc. Now, extending out from the embryo, you have the amnion, and within that, the amniotic cavity. Okay, next thing that happens is gastrulation. This is a very critical period of development, and it occurs during the 3rd week. And here the epiblast is going to form 3 layers. And we call these the primary germ layers. And the reason we call them that is because this is where cells from all the body tissues are going to develop. And these 3 primary germ layers are the ectoderm, the mesoderm, and the endoderm. And this 3-layered structure is now going to be called an embryo. All right, I'm going to show you a short video that goes over the process of gastrulation. All right, so we said gastrulation is going to begin with the formation of that primitive streak, which is basically just a thin depression on the surface of the epiblast. The primitive streak is going to then contain the primitive node, which is at the cephalic end of the streak, and it consists of an elevated area surrounding a depression called the primitive pit. Then invagination is going to occur. Here, cells are going to detach from that epiblast layer and sort of penetrate down, migrate down through the primitive streak and lodge between the epiblast and hypoblast layers. The epiblast is going to be the source of all 3 primary germ layers. So all body tissues and all organs are going to be derived from these layers. The endoderm, remember this is going to be when those epiblast cells displace the hypoblast. The video called it the definitive endoderm. The mesoderm, this is that new primary germ layer of cells that was formed by the invaginating epiblast cells. And then, of course, the ectoderm, which are the cells that remain in the epiblast. All right, here we're looking at the primitive streak in gastrulation. So here is the cephalic end. Here is the caudal end. Here is the primitive streak. Here is the primitive node containing the primitive pit. And you can see these cells from the epiblasts are going to kind of dive down in through this primitive streak. And they're going to invaginate and move and displace the hypoblast and then wedge in between the hypoblast and the epiblast, forming the endoderm and the mesoderm. All right, now let's talk about folding of that embryonic disc we just formed. So in the late 3rd and 4th weeks, it's going to start to fold in on itself. And there's 2 types of folding that occur. There's cephalocaudal folding and transverse folding. Cephalocaudal folding is going to occur in the cephalic and caudal regions of the embryo. So here we're looking at the embryo. This is the cephalic end. This is the caudal end. What you see is rapid growth of the embryonic disc and the amnion, and no growth of the yolk sac. Here you can see the amnion growing and the embryonic disc growing rapidly, and they kind of wrap around this way. And this causes the head and the tail sort of fold in on themselves. Next, you get transverse folding. So here, the left and right sides of the embryo are going to kind of dive down and curve back towards the midline. And they start to pinch off the yolk sac. You get fusing of the sides of the embryonic disc in the midline, and this is going to create a cylindrical embryo. Thanks to transverse folding, you get ectoderm solely on the exterior of the embryo, and you're going to get endoderm solely on the interior of the embryo. The yolk sac is going to be pinched off from most of the endoderm except through the vitelline duct, and this is going to create the torso region. Now, some of that ectoderm on the dorsal side of the cylindrical embryo is going to differentiate, and it forms special nervous system tissue. We call this neurulation. So not only is the ectoderm going to form the epidermis, but because of the differentiation of the ectoderm, it's also going to form sense organs, the pituitary gland, the adrenal medulla, the enamel of the teeth, and even the lens of the eye. Differentiation of the mesoderm. So there's 5 categories of mesoderm. There's the notochord. This is formed by a tightly packed midline group of mesodermal cells. It's the basis for the central body axis and the axial skeleton. And the notochord actually induces formation of the neural tube. Then there's the paraxial mesoderm. This is found on both sides of the neural tube and forms somites, which are kind of these repeating blocks of tissue. So these form the axial skeleton, the muscles, the cartilage, the dermis, and the connective tissues. There's the intermediate mesoderm. This is lateral to the paraxial mesoderm, and it's going to form most of the kidneys, the ureters, and the reproductive system. There's the lateral plate mesoderm. This is the most lateral of the layers of the mesoderm. It's going to form the spleen, the adrenal cortex, and the cardiovascular system, as well as serous membranes and connective tissue of limbs. And then you have the head mesenchyme. This is going to form the connective tissues and the musculature of the face. All right, so kind of working inwards to outwards, you have the notochord here. Then you have the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. Okay, and then what happens to the endoderm? Well, it's going to become the innermost tissue after that transverse folding, and it's going to end up forming the linings of your GI, respiratory, urinary, and reproductive tracts. So if you hear the word epithelium, you need to be thinking endoderm. It's also going to form the tympanic cavity and the auditory tube, the liver, the gallbladder, the pancreas, the palatine tonsils, the thyroid, the parathyroid glands, and the thymus as well. Next, we enter organogenesis. This is organ development, and it begins once the layers have formed and have completed their folding. By week 8, you're going to see upper and lower limbs have their adult shape, and most organ systems are going to have at least a rudimentary form. The developing organism is going to be particularly sensitive to teratogens during this time, and teratogens are just substances that can cause birth defects or death in the fetus, and they include things like alcohol, tobacco, drugs, and even some viruses. So what's happening in organogenesis? Well, this is the peak development period. It's the time where most organization and construction of the body's organs occurs. Now, it's going to vary for different body structures. For example, limbs have a peak development period between weeks 4 to 8. Teratogens are going to be the most dangerous during the peak development of a particular system. So if you have a teratogen that would harm the limbs, it would be most likely to do this between weeks 4 to 8. Then finally, after week 8, we're out of the embryonic period and we enter the fetal period. So this is from the beginning of the 3rd month until birth. And what you see here is maturation of those tissues and organs, rapid growth of the body. You see the length of the fetus is measured as crown rump length or crown heel length. And a 2.5 centimeter embryo is going to grow to an average of 53 centimeters at birth. And so that's like going from a 1-inch organism to a 20-inch organism. And the weight increase of the fetus is going to be most striking during the last 2 months of pregnancy.